Welcome to the Gothenburg Natural History Museum and tonight's event. We are part of Västlandet and the region of Västra Götaland, and we do this in support from Folkuniversitetet in Gothenburg. And tonight's event is also a collaboration with the GGBC, the Gothenburg Global Biodiversity Centre. We are going to film this event, and you can see the camera down there, so if you don't want to be on camera, just please avoid the aisle. And if you have any questions for our speakers and you don't want to say them out loud, you can just tell them to me and I'll be on camera. So you don't have to be there if you don't want to. Or you can just do a little dance and be on camera as well. We're going to put that out on the website, so I think you'll have a bit of an audience. But I'm just going to leave Alison before I say something stupid. Welcome Alison and Louise, Emke and Anne-Sophie. And you'll get a little bit of an introduction from Alison.
Yes, thank you, Renee. Yes, so thank you everyone for coming. This feels really loud. Is it okay? Okay. I don't like having things so close to my face. Yes, so I'm Alison Perigal. I'm the director of the Gothenburg Global Biodiversity Centre. It is really loud, isn't it? It's not just my head. Can I use that one? No, okay. Better? So I'm going to say just a couple words about the Biodiversity Centre before we get into the talks, just so that everyone can know a little bit about what we are and what our goals are. So I think, yes. The most important thing to know about the Biodiversity Centre is we are basically a network. So we're 13 different organizations together working to promote research and outreach in biodiversity. So the Natural History Museum is one of our 13 partners, and it's really nice that we've started organizing events here. And they are giving us a channel for some of our researchers who you'll hear speaking today to present their work to a greater public. In addition to the Natural History Museum, we have a number of other public partners, including Botaniska, the botanical garden, the Sjöfartsmuseet, the aquarium, which is currently under renovation, Havets Hus, also under renovation. We're going to have some really good biodiversity places in about a year when everything's back up and running. Universium, which is not under renovation, and in Nordens Ark as well, up in Hunebostrand, up a couple hours north of here. Additionally, we have partners at Chalmers and the University of Gothenburg. Are you going to go? No? No? Yeah, there we go. So that obviously doesn't add up to 13 partners. Our 13 partners include all of these different departments at the University of Gothenburg, as well as one at Chalmers. So that's how we cover both the research and the outreach side of things. And our goal, as I said, is to promote research and outreach in Western Sweden, because we have this huge amount of biodiversity here. We have a huge, huge, huge amount of research that's going on both on things around here and globally. So we have very different perspectives from very, very different types of researchers and people. And so we want to create a platform where we can bring the researchers from these different departments and from these different organizations together to make sure that our research can be as good as possible. And our main goal here is within the UN's Agenda 2030. We're trying to address the goals of the two Sustainable Development Goals, 14 and 15, life below water and life on land. So, sorry, this thing's driving me nuts. Yeah, so a lot of our research is playing into that and how we can make sure that the public has the information available to them in order to reach these goals, which are fairly complex but are based in fundamental research. And with that very short introduction, I will hand it over to our speakers because they're the ones that you're here to listen to. And our first speaker this evening is going to be Anne-Sophie. She's a PhD student at the Department of Biological and Environmental Sciences at the University of Gothenburg, and she is doing a project in a greater project funded by a European program called Plant ID, where it's got 15 different students at universities in different countries around Europe, and they're all doing projects that work with molecular identification of plants in one way or another. So, welcome, Anne-Sophie. Yes, and now you get to figure out how to put this on.
Yes, I'm trying.
I'll lick it first.
Yeah. Thanks.
Yeah, you should do your dancing.
Oh, yeah.
Voila. Forward,
Okay.
backwards, and a laser.
Okay, perfect. So hi everyone, and thank you for being here. So as Alison said, I'm Anne-Sophie Quatela, and I'm going to talk about how my work is linked to biodiversity and what I did to end up doing that. So first, I guess you all heard about biodiversity linked to conservation, especially in this context of climate change. We all hear about the importance to conserve endangered species. But conservation is not the only field linked to biodiversity. What I do, my field, is about genomics, so the study of genome architecture and evolutionary biology. So I'm a PhD student at Gothenburg University. I started a year ago. As you can hear, I'm French. I'm from Grenoble, here. So I did my study at Grenoble University with a bachelor degree in general biology, more focused on ecology, not that much on evolution. And then I did a master degree in ecology, biology, environment. So in the continuity with my bachelor degree. So what I'm focusing on now, so I'm a plant biologist, as I said, an evolutionary biologist. And what I do basically, it's taxonomy and species delimitation. So it's the science that classifies organisms. In taxonomy, it's based on morphological criteria, and in species delimitation, we work with DNA. So our criteria are DNA. My study model, it's Silene. So you have just 2 examples of species, but there are many more. It's an important field for the society. First, taxonomy and species delimitation. It's the basis of biology. You can't study species if you can describe and classify. So my study model has a little particularity. It's a particularity that concerns these items. That might be surprising, but French fries, cigarettes, some shirts and coffees share what they are concerned about what I'm studying here. Because French fries are from potatoes, cigarettes contain tobacco with other many bad things, some shirts are made of cotton, and coffee beans. So they all have, to be very simplistic, they have more DNA than expected. It's called polyploidy. You don't have to keep that term in mind, but it's a specificity that make classification on a genetic point of view much more complex. So that makes my job a bit more complex, but more interesting. So that's the main challenge of my PhD. So now I explained what is the link of my project with biodiversity. So I'm going to explain what I did to end up in this PhD. So after my bachelor degree, I was not confused, but I wasn't sure about what I wanted to focus on in master degree. It's good to have a specialty. So I decided to take a year off to do some voluntary internships. And I did 2 internships in Palenque, in Mexico. So it's a famous archaeological site for this Maya site. Probably heard about that. Well, yeah. So I did 2 internships. One on Aramaceo, this parrot, based on focus on conservation. There is a kind of zoo there that try to rehabilitate many different species, and they have partnership with the University of Mexico. So they have they have projects more focused on research, like population genetics. So I did a very basic things. It wasn't a research project, but you have to get your hands dirty sometimes. So then I did a project on ethology, on howler monkeys. So ethology is a science that studies animal behavior. So how you can guess howler monkeys howl. They have vocalization. So I was the assistant of a master student that was trying to understand the meaning of the vocalization. So it was a lot of field work, following a group of monkeys around Palenque. And we were just, recording, taking notes. Well, I have a video about monkeys howling. It's, yeah. If I manage, yeah. You can see a baby here. It's a family. Well, I think it's okay now. Yeah, bon. So after these 2 internships in Mexico, bon, I had a small overview of 2 possibilities linked to biodiversity. It was time to go back to Grenoble to do my master degree. Yeah, so I studied a bit, as I said, ethology, conservation, but it's just 2 possibilities in biodiversity. And I had this opportunity at Grenoble Alpes University, so my university, to study evolution on plants, the first year of my master degree. So very short internship, 7 weeks, on Espelecia, these plants in the Andes, in a very specific ecosystem called Paramos. So it's an alpine ecosystem in a tropical environment. And I was studying their diversification, which factors influence their diversification, because it's a very diversified group, Espelecia. It's been a trigger in my career, because it's when I really decided to study evolution. So of course, you compare with your other experiences, and for the very first time, I was absolutely convinced that I wanted to do evolutionary biology on plants. So I just decided to keep going. And thanks to contact from this lab I was talking about, I've been able to go to South Africa for my master thesis, so the second year, for 6 months in Stellenbosch, up here. It's the wineland of South Africa, so if you drink wine from South Africa, it's probably from Stellenbosch or around. So what did I do there? I studied aloes diversification, the influence of their height on their diversification. It was a very theoretical work, like for the previous one I just talked about. But I've been able to do some field work for my supervisor there on another group. So they are succulent plants. It means they are adapted to a dry environment. So they keep water. You can see a very thick leaves because they keep water. So the point of this project was to sample, to retrace their evolutionary history. So to do some genomics. It was here in the Knersflakte. It's this landscape that looks like the moon. It seems like there is nothing there, but you can see many plants looking like this one, that we can call stone plants. It's an amazing area because, well, it's protected, so it's not easy to go there. So when you have the opportunity, you go. And for the plant I just talked about, they can have water mainly from the fog in the morning by absorbing this humidity. So that's basically, what I did. And I want to illustrate one of the many advantages to do fieldwork. Like, we saw many different flora species, but fauna as well. So this is an aardvark, an ant eaters. Normally, they go out in the evening, early evening. It's not easy to see there. I met many Africans who never saw any aardvark, but when you do fieldwork for a while, you are lucky and you can see amazing species. So do fieldwork if you have the opportunity. So it's the end almost of my talk. So after these two theses about evolutionary biology, I just wanted to keep going in that field. And to study that, you go into research in general. So I found this opportunity here in the project Alison talked about, Plant ID. And that's how I ended up to Gothenburg. So I just have two take-home messages. Just want to emphasize that biodiversity involves many different fields, and in one field there are many different steps. Fieldwork, lab work, but a lot of analysis as well, so it's not just the funny part. And another message, it's important to not always choose the easy path. Don't hesitate to make some breaks to identify what you really want to do. That's all. Thank you for your attention.
A couple of questions now. Anyone has anything for Anne-Sophie?
We can talk after if you want. We'll be around.
I want to know about the howler monkeys.
Okay.
Is there actually, like, are there different sounds for this thing?
Yes. Actually, the male, the alpha male howl mainly, and it's so in this area there were several groups, all with a familial structure. So it's mainly to mark their territory. I don't know if it's English, but it's mainly like to impress the other group and to give some information about the location as well. So that was the conclusion of this master thesis. Yeah.
Do the females howl sometimes as well, then?
No. But the young male does it. I think it's mainly for, anatomical question. Yeah, for anatomical reasons, because the males have this big throat, not the females. Yeah.
Thank you for this interesting talk. I was wondering about the elevate plants. How tall do they get? And they get taller when they get older. Is it measuring their age?
No, actually, you have some species that are genetically very tall, like tree-like. This one is several meters high. So it's genetic. Some of them, like this one, it can be called a grass aloe. They won't be more than a few centimeters tall. So, of course, when they get older, the ones that are genetically shaped for that will get taller, but otherwise, no.
Great. All right, thank you.
Thank you.
think we can swap and present Emke will be next. Emke is halfway through her PhD right now. She's working at environmental systems analysis in Chalmers. And she has an extremely collaborative PhD. So it's between, let's see if I can remember all these, Chalmers, SLU, Quatela University, and University of Gothenburg, right? So she'll tell us a little bit about what she's doing.
Yes. All right. Well, nice to see you all here. My PhD work is about biodiversity and life cycle assessments. And before I will tell you something about what it is, I will give you a little bit of an introduction of my background. I'm from the Netherlands, a small town called Castricum, close to Alkmaar. And I studied biology at Leiden University. Both are, if you've got the map of Holland, in the, well, the underwater part of Holland right now. And I studied biology because I wanted to be a biology teacher. And, well, I can't say that I wasn't, like, attracted by the many, what is it, holiday days you get as a teacher, at least in Holland. But anyway, I studied and I became a biology teacher. But when I was in front of the classroom, I thought, like, yeah, but I'm not really ready for this. I'm not really finished studying yet. So I want to, you know, develop myself a little bit more first. So instead, I went studying again, and now it was industrial ecology. And industrial ecology studies industrial systems and tries to minimize their environmental impact. It's called industrial ecology because it conceptualizes man-made industrial systems as natural systems. Or the idea is that they work in the same way as natural systems. And natural systems are very efficient. I mean, the waste and the byproducts from one system are used as input for other systems. So what if we can do the same for industrial processes? Minimizing the natural resource use, no waste. And this idea, this is also called like the circular economy, would be a sort of ideal form of a sustainable world. But we're not there yet. I mean, the awareness is rising. There are more and more companies who are trying to minimize this, the impact of their products. Policymakers are trying to make decisions which leads to, you know, less waste, for example. There are all kind of eco-labels around. Environmental footprints, maybe you've heard of the carbon footprint, for example. And these kind of footprints are based on life cycle assessments. So basically what a life cycle assessment is that it's a tool to assess the environmental impact of a product or a production system. And this is quite important because consumer behavior, like how we consume products, and mainly food products actually, are one of the leading drivers of environmental impacts. So it is important to try to minimize their impact and by looking at the supply chain and see, well, where can we actually do something what matters. And this is more and more integrated in national policies, but also on the European Union level. Life cycle assessment becomes more and more important. The European Union also has integrated policy. What is it? Integrated product policy, and with that they try to influence consumer behavior to buy more greener products. But And yeah, well, it's not only about calculating like the impact on climate change. There's much more in this. Eutrophication, acidification, how much, if there are any substances maybe which can cause cancer, all these kind of problems can be assessed with life cycle assessment. But I don't see any biodiversity here. And this is quite problematic as it is. I mean, it's such an important decision support tool. It's used more and more. And we know that biodiversity is important. We know that we sort of depend on biodiversity for clean drinking water, for oxygen, for pollination, for all these kinds of services. We need biodiversity. So we want product developers also like develop products which are good for biodiversity and not only for the other problems. But first, why? I mean, why do we want to consider the whole life cycle? And life cycle assessment is assessing in the whole of life environmental impacts. And if I say whole of life, then you know, I can sit down with you and I can ask you, like, tell me something about your life. When did your parents meet? Which year are you born? What did you do when you were 5? Where did you live when you were 10 years old? And you can even tell me if I ask you, like, what do you want to be? Where do you want to be in 10 or 20 years? You could tell me, like, well, you know, this is how I want my future to be. Now imagine doing that for a product. Like, you take this laptop and well, I say, well, tell me this life story of this laptop. So LCA gives you sort of the product life of the laptop. And by doing so, you sort of more effectively can point out which steps of the life cycle of this computer causes environmental problems. And we are mainly doing that because we want to prevent a problem shift. And with a problem shift, I mean that maybe we think we came up with like a great alternative, a very nice, sustainable thing, but we mess up something else. In the case of electric cars, for example, it's not very clean to drive an electric car if the electricity is still produced by burning fossil fuels. It was actually yesterday on the radio, an interview with someone who said, like, you know, in some countries it's even better to drive a diesel car than an electric car because the electricity production is so bad in those countries. Same for biofuel production or some kind of biofuel production. It's not much cleaner if there's a lot of pesticides involved, for example, which are really bad for the environment. So you want to eliminate this kind of problem shifts. You don't want to, you know, end up with a problem somewhere else, and you know you forgot to think about that. Now, let me give you an example of such a production chain for beef production. To produce beef, you need some pasture lands for the cows to graze on. Silage is something what the cows eat, a lot of it actually. They put it in those bales because then it starts to ferment, it's called. Easier to digest. Some mixed grains and some soybeans, which could be like produced in Brazil, for example. So if you're looking at the whole production system, it looks something like this, but it's very simplified. You have to think that, like this plastic, for example, is also produced somewhere. The soybeans should be transported, which also causes environmental problems or is an environmental burden. Yes, so in reality there's more involved in this. And, you know, these raw materials are also produced somewhere. So in the end, you end up with something which looks more like this. Oh, I'm going too fast now. So it could be humans are very good in making complex systems as well. So if we sort of sum up everything which goes in and goes out from this product chain, we could see that for the production of beef, roughly 40 square meters of natural grasslands are used in Sweden, 25 square meters of arable land in Sweden, and then you've got some square meters of arable land in Brazil. Now, these square meters of land use doesn't tell us a lot about biodiversity, so we need some kind of model which sort of characterizes these square meters in an impact on biodiversity. And these models are quite complex. And yeah, there's a lot of uncertainty involved here. But what they have in common, these models, is that they all consider like two different kind of land use impacts on biodiversity, which is the transformation impact and the occupation impact. So imagine that you've got a forest. This forest is cut down. You've got some agricultural field instead. This is, you know, they use this agricultural field for a while, and when it's not needed anymore, well, land abandonment is not very common, but it happens. The land can sort of regenerate and reaches a new steady state. Now, the total impact on biodiversity is now this this area. Like this drop in quality compared to sort of the original state in time and then in space, which are the amount of square meters. You can imagine that, I mean, there's a lot to say about this as well. A natural state, I mean, if you've got an agricultural field, it's maybe used for like 100 or 200 years. So we don't even know what it was like before. And even if we try to reconstruct what it was before, we might fail. It's a difficult measurement. Then we also have the question like, yeah, but how do we measure biodiversity then? There are so many indicators. Which one to pick? And this kind of problems really, it depends a lot on the modeler. And that's something I would like to give you as well, is that each model we use to calculate the impact on biodiversity is some kind of construct of the modeler. The modeler made so many choices in how they're going to measure biodiversity, what they think biodiversity means, that we have to be a little bit careful using those models, because, you know, it gives a really good indication of the impact on biodiversity, but it's not the truth. And that's important to keep in mind. But still, we think it's important that decision makers have some kind of measurement or have some kind of number they can use to steer their policy making into sort of a direction which would be beneficial for biodiversity. All right, thanks a lot.
Do we have any questions? I have one. How can these analyses be meaningful then? In what way can they be used if it's so much ambiguity? How can policymakers or that type of people actually take them and say something and make decisions from them?
I think it's a good start, and I mean, policymakers, you know, the best they could get is just one number for biodiversity, and you know that there's a product and there are two products, and one has a score of 10 for biodiversity and one has a score of 5 for biodiversity. And that would be ideal, but it doesn't work like that, obviously. But life cycle assessment still tries to do that. And in one way it's good because, you know, one measurement is better than nothing, and if they can take some biodiversity with them into policy making, it's good. But if we really having some local problems, local biodiversity problems, then LCA might not be the good tool for it. So we have to be very careful in when we use LCA and for what purposes. That not the LCA is leading all the decisions towards, like, yeah, I don't know, different measurements in the field.
Thank you. It's definitely important to be able to explain in some degree relative the relative contribution. It's really something we're missing a lot in biodiversity. I think it was really interesting when you showed that no one has actually done this for biodiversity at all.
No.
You're really
no, there are some people who are doing this and modeling. Yeah, but I mean, biodiversity is a broad concept, so only with measurement for species richness, we are not there yet. We want to take into account more types of biodiversity because we know it's more. It's also on a genetic level, it's on a landscape level, and then you've got completely other scores for biodiversity. Yeah.
Any other questions? All right. Thank you.
All right.
We'll bring Louise up now. So Louise is Louise is our third speaker. She's our furthest along in her career of our three early career researchers. She's her position is called researcher, so it's a position after a PhD. And she works in the Department of Earth Sciences at the University of Gothenburg. And she's working with climate change and how that interacts with biodiversity.
Thank you. How is this? It's a bit too loud. Good. Well, thank you for being here for these talks. And this is the last talk. Yes, and as Alison said, I'm going to talk about climate change effects on ecosystems. So just to introduce you to what an ecosystem is, I have a little definition here for you. An ecosystem, it's a community of living organisms and the non-living components in their environment, and these things are interacting as a system. So that is what an ecosystem is. And the focus here today is on grasslands. We've already seen some lovely pictures from grasslands. And if you take the broadest possible term for grasslands, these occupy about a third of the land surface. So grasslands are super important ecosystem types. Just a little bit about myself. Yes, as Alison said, I already did my PhD degree in Denmark some years ago. And since then, I've been on a track of employment through Europe. And it's just been super interesting to work with climate change experiments in the field in Denmark and the Netherlands and in Germany and lately also in Australia. So now I work as a researcher here in Gothenburg, and I'm interested in nutrients in soils and in plants and how effects from climate change spread in an ecosystem. So in this talk, I will just take you through these points. Something about climate change. You probably all know the greenhouse effect. I have something to tell you about the field experiments we make out there in the field. And then I will show you actually data, some data slides of how plants react differently to climate change. I will take this to a global perspective and give you some take-home messages. So I hope I can keep you all interested even through these data slides later on. So here's the first one. This is measurements of greenhouse gases. This is actually real measurements. The points are concentrations measured in air bubbles in ice cores. So the green color indicates CO2 concentration. The orange is methane, and the red one is nitrous oxide or laughing gas. And these are all important greenhouse gases, even though the scale here is 1,000 times upscale compared to CO2. Sorry, this way, yeah. So the global warming potential of these greenhouse gases, it's not the same. So if you have CO2 set to 1, here you can see that nitrous oxide is actually 62 times as bad as CO2. It gives more warming effect, and methane is almost 300 times as bad as CO2 in the atmosphere. So that's why we also consider these other two greenhouse gases. So you can always look up what is the daily concentration in the atmosphere of CO2 on this web page here. Last time I looked, it was 414 ppm. So that's a number that's always increasing. So you can always follow that. I have actually a little cartoon of the greenhouse effect that we can see after my talk if you are interested in this. But what happens during the greenhouse effect is that the globe gets warmer. These two examples of model worlds show temperature increase as it could be at the end of our century. It's two different examples. So it is change in temperature, and if you have a very red color, it gets very much warmer. The yellow colors are less warm, but as you can see, there are no blue colors in these pictures. So there are no predictions that indicate a lower temperature in the future. So this is what modelers do. So those are my colleagues. This is not exactly what I work on. There are also being predictions made on how the rainfall pattern will change. So these two model examples show you how the rainfall pattern can change in the future in two different examples. The very blue color here would give you a higher rainfall amount every year, and the yellow and reddish colors, they would give you drought, so less rainfall. So yeah, those are different versions of the future. And this is what keeps us interested in what the effects actually are of these climate change factors. So now I come to show you something from field experiments. So this is actually a field experiment with elevated CO2 directly on this grassland. So the research question behind this is, what are the effects of elevated atmospheric CO2 on the managed grassland? So this is a type of field experiment where air is circulated. So we have elevated CO2 air being pushed out from this tubing system over the plot. And then there is another part of the tubing system that sucks the air back. So air is circulated, and this is now air with elevated CO2 concentration. This field experiment is in Germany, so not so far from Frankfurt, as you can see. This is an aerial photograph. It's a bit warmer region than here, and the precipitation is about what we have here in south of Sweden. You can see it's circular plots, so these are the plots that you saw from the side. And on this field side here, there are 3 rings with elevated CO2, and they get their supply of CO2 from a CO2 tank over here in the corner, and then it's channeled through some tubing system out to each ring. And there are 3 rings with normal air being circulated. So those are the controlled plots. So what are we actually measuring in these plots on the grassland? We are measuring biomass. So biomass, this is the weight of the plant that we harvest. And then we want to measure the response to elevated CO2. So this is actually how much more, for instance, biomass we get in the CO2 treatment relative to the normal. And now we're going to make a calculation. So we take the amount in elevated CO2 plots, we subtract what we have in normal plots, and we take this relative to normal plots. We can take it as a percentage, and then we have a response percent. So now I show you data with response percent, so response in biomass to elevated CO2 treatment. So what we have here is a response scale. So if you just can see the blue line that goes through 0, this is the moment when there is no response. So anything above this line is a positive response to CO2, so higher biomass under CO2. And if you're below the line with a negative number, you have a negative response to CO2. So the harvesting on this field site, it has continued since 1993 and up to 2014 in this graph here. And in the beginning, they just harvested everything, but then in 1996, they started to divide it into grasses and herbs as 2 different portions of the harvest. And you can see the red dots, this is grass, and the green triangles, that's herbs. And you can see they are responding differently. And we could talk about this data set in 3 sections. So we have an initial phase, we have a short-term phase, and then a long-term phase as the last years. And if we just look at the initial and the short-term, you can see that the response in grasses and herbs is super different. Grasses are positive, herbs are negative. But if you then move over to the long term, you can see that grasses and herbs, they then find the same positive response pattern. So they act differently. So just to give you a feeling for how much we harvest here, this is about 700 grams biomass per square meter per year. So that's the harvest amount. So we have this permanent managed grassland with 18 years of data from this phase experiment. So why is the vegetation responding like this? Yes, so we have this response that is connected between the plant leaf and the soil. So elevated CO2 actually gives a positive effect on soil moisture. And this is due to less transpiration from the plants. They have a better water household. And this is something that happens at the leaf level. So you can see this leaf and a small subsection of the leaf. And the leaf has got these small holes called stomata. So this is a cell, many cells on the leaf surface. And those cells, they can open more or close more. And they are these holes that let in CO2, but they also let water escape or oxygen escape. So when you have an atmosphere around this leaf with a higher amount of CO2, the plant doesn't have to have such an open hole. It can close it a little bit more, and this will then let the plant save water. So you have more CO2, the plant saves water by closing those stomata. So this is why this whole response through the plant down to the soil, where the plant is getting its water, it gives a better water household under elevated CO2. And this is also what we saw at this field site as expected. In this long-term situation, the soil moisture was higher under elevated CO2. But then on the short term, the soil was then contrasting to what we could think from this concept here. The soil moisture was actually worse off. And we think that this was because of a competition situation between the grasses and the herbs. So the grasses, they are just pushing on. They're responding positively to CO2 from the beginning. And they are growing so much biomass that they're actually drying out the soil. And this has a negative consequence for herbs. But through the years, they catch up and then also start to have a positive response. So the short-term result in this experiment does not predict the long-term result at all. So herbs and grasses, they have different response to CO2, and it was linked also with other local climatic and abiotic effects. So there were some effects from early frosts in some years that really was difficult for herbs to handle, and they were set back again in some particular years. So there are more climatic factors than just water. So now I took this data from this one grassland in Germany and joined it into a big meta-analysis of many grasslands worldwide. So now we have here data from, I think it is 26 different grassland experiments with elevated CO2. And we're looking on explanations for the response. And it turned out that the rain seasonality was very determining for the response in grasslands. You have 2 graphs here. On the y-axis, you have again a response percentage, and the 0 line. So when you have positive number, you get this positive response, and it can be smaller and smaller and even negative. And the first graph closest to me here, this shows on the x-axis mean spring precipitation. So how much rain you have during spring, the main growing season, when everything starts. And it turned out that as you have different ecosystems with more and more spring rain, you also have a more and more positive response to the CO2. And this was a bit difficult for some to understand why this could be, but I think it can be understood from the nutrient side. So this moment when the grow really starts during spring, this is also where the plants need a lot of nutrients. So if you have more rain, more nutrients are mobilized in the soil. They can be used by plants and they can grow more biomass because of this nutrient supply. And then there is the other half of the graph, which is the response axis up and down. And then on the x-axis, you have the mean non-spring rain, so the rain during the rest of the year. But here it's another response. So if you have field sites with more rain during the rest of the year, you have less response to CO2. So actually those sites that have less rain during the year, except the spring, they have the highest response to elevated CO2. And this is well connected with our theory because this elevated CO2, this actually helps the plants save water. So this is the whole thing behind these two different sets of results. So this was, the dots are from real field site measurements, and then there is these regression lines made. But now the group of researchers, they upscaled this to global scale. So they gathered information about rainfall patterns on all the grassland ecosystems they could find, and then colored the map with how much biomass response we would get. So you can see the color scale here, the green and very blue colors. This is where the grasslands are responding very positive. And the more red and orange and very red color, this is where grasslands actually respond negative. So there are areas, regions, where you will get a negative growth under elevated CO2 at grasslands. And it's quite substantially big areas. So, yeah, so is this good or bad? I have some take-home messages for you here. First, you have to realize that there are some limitations on these climate field experiments, such as their duration, because they are very expensive to run. You could also argue that you are making a pollution with elevated CO2 treatments, just like that on the field. But they're expensive to run, and normally they are set for a few years only. And we saw from the example with grasses and herbs that you have first some negative responses in the first years that could become positive. And this could be variable between groups of plants. We also found in a different meta-analysis, some examples where we have a very positive response building up, becoming more and more positive, and then after many years, after 15, 17 years, then this response in biomass became smaller again. So we always need longer experiments to actually know more about how responses are. So the point from the meta-analysis that I showed you is that the rainfall pattern is really important for the elevated CO2 response. And there are regions with less growth under elevated CO2. And if this phenomenon becomes widespread, it has very negative consequences for the feeding chain, for the cows. And of course, less growth in biomass also has a consequence of less carbon uptake in the vegetation. So, but on the good side, I would say that higher diversity in grasslands, this keeps a higher potential for positive responses because there can be groups that can take over if other groups of species are reduced. So if we have ecosystems that grow more plant biomass under elevated CO2, then this can help us to draw CO2 out of the atmosphere as a good feedback. So yes, with this said, I thank you for your attention, and do you have any questions?
Thank you. Questions for Louise? Yes.
Thanks. That was a really interesting experiment with the elevated CO2.
Yeah.
But I was thinking, you only looked at the CO2 in that case, whereas with climate change, of course, due to the CO2, the temperature would increase as well.
Yes.
So is there any way that you can incorporate that into these kind of experiments?
Yeah, that's a really good question, because you don't only have elevated CO2, you also have a temperature increase, or you maybe have changes in rain, and yeah, there are many factors. And there are actually quite a few experiments that combine treatments, so they both have CO2 but also a warming on top of this. And then normally you also have only CO2 and only warming and control plots. So this is how to test this experimentally, that you make more treatments in the field.
Do you get the same results?
This is a super interesting question, and no, you often have strange responses when you combine treatments. It is not so seldomly seen that you can have a positive response in CO2 treatments and a positive response in warming treatments. And then when you combine these, you get no response. And this is things that always drive researchers crazy. Why is 2 and 2 not 4? It's 0 instead. So yeah, there's research enough for many years there to figure that out.
Any other questions?
Are there any computer models that predict this, like very well, like what is the optimum CO2 level that one can reach to have the maximum biomass to be produced in a particular region of rainfall or something?
Yeah, I think I understand your question. You can imagine that you can just keep on growing more and more biomass if you get higher and higher CO2, but there is an upper limit, but I can't tell you what it is. But I have seen predictions that when we reach a certain CO2 level, you will not get more growth. So there's sort of a roof. But sorry, it's not my job really.
Thank you for a fantastic lecture. My question is, there is a study by University and Chalmers that called Gothenburg 2050. This has been done in beginning of 2005. Do you have any response from this study?
I don't know what you talk about. What do you mean? Sorry. Can you explain it a little bit more?
There is a research.
Yeah.
Result being done by the University of Gothenburg and Chalmers, and between 2000 and 2005. It's called Gothenburg 2050.
I don't know this.
You don't
No,
know?
I don't know this. Maybe you...
I think this is an environment contour in Carl Johans.
I'm not familiar with that either.
No.
This is studies by Municipal Corporation of Gothenburg. They collaborate between these two universities to do for Gothenburg 2050. Because which I have listened, you are having a project also 2081, 2100. That was then.
Ah, that's the models I can...
Between these two.
You
Yes.
mean these models, yeah. Mm?
Yes. Between 2005 to 2081, there is a study in Gothenburg. I don't know if you can find from libraries, some of the university, if you ask, you can take part of this study.
Okay, yeah, I can try to look it up.
Yes.
Yeah. Mm?
This was
Thank you.
Thank you for that.
Any other questions? All right. Well, I think we can thank our speakers again, and we will have a little mingle afterwards. You can ask questions if you'd like to come up and chat with them. We'll be around for a little bit.
Yeah, I think we have to say something about our other joint events.
Yes. Let's thank our speakers first.
Oh yes, we can see that. Do we have time?
That's right. Yes. Let's see.
Yeah, you can.
Do you know what the greenhouse effect is?
I just need to change the settings.
All right, in the meantime, I can mention that we have another event together, the Natural History Museum and the GGBC, and that's going to be on May 22nd for Biodiversity Day. We have lots of events going on at the GGBC, and it will culminate here in the evening. I'm so loud already, though. It will culminate here in the evening. We're going to have an event called Tales from the Field, and it's going to be stories about field work, gone right and wrong. Probably more wrong than right.
Mostly wrong.
Mostly wrong.
And crazy.
And this should be a really fun event about we heard a little bit about the cool field work that people do, but this was what happens when it goes right. And when you get scientists going over a couple of years, they have some really funny stories about when it goes wrong. So that's what we're going to share in honor of Biodiversity Day.
Yeah, thank you.
Do you know what the greenhouse effect is? It is the natural phenomenon that allows our planet to maintain the necessary conditions to harbour life. The atmosphere captures some of the sun's rays that reach the Earth's crust, keeping them inside to get an average temperature of 15 degrees Celsius. If the atmosphere did not catch any of these rays that bounce off the surface, the average temperature of the Earth would be minus 18 degrees Celsius. The atmosphere is composed of various natural gases, of which nitrogen, oxygen and argon constitute 99.93%. However, there are other gases such as water vapor, carbon dioxide, methane, nitrous oxide and ozone which have a greater impact on the greenhouse effect. Moreover, artificial gases called chlorofluorocarbons are also present in the atmosphere. In the right proportion, these gases perform their duty, but when its concentration increases by the action of man, the atmosphere retains more heat than necessary, causing rising temperatures, melting ice caps, desertification, fires, storms and floods. Why do greenhouse gases increase? There are three main reasons. The burning of fossil fuels like oil, coal and natural gas, forest deforestation and destruction of marine ecosystems that absorb carbon dioxide, and the increase of a population that increasingly consumes natural resources. Now that you know what causes the problem, you can help reduce the emission of greenhouse gases. How do you fight climate change?
Yeah. Thank you. That was nice.
Very happy music.
Yes, thank you so much.
Thank you, everyone.
And yeah, if you have any questions that you didn't think of before, or that you didn't want to take in front of everyone, so we'll have a couple of minutes, a little time to talk to each other. Thank you.